So my name is Matthew Phillips, neurologist here at Waikato. I'm going to be discussing a topic of metabolic strategies in healthcare, a new era, and I hope after this half hour you'll understand what I mean by a metabolic strategy and why I think there's a new era in medicine approaching over the ensuing decades. I'm going to focus exclusively on Waikato research for this talk. So every study you see has come out of this hospital. We go back 200 years to Louis Pasteur. Louis Pasteur was a hero of mine when I was a kid. Yes, I didn't like Superman so much. I thought Louis Pasteur was much cooler. He was a French chemist and microbiologist who championed germ theory, which is a theory we still use today very strongly. Germ theory is a disease-centric theory. It emphasizes pathogens as the origin of disease, and of course, in Pasteur's time, it was applied to infectious disease. Pathogens had to be targeted and eliminated to clear the disease. It was quite straightforward and very successful at the time for various reasons. But the focus of this theory is on suppression and elimination. Pasteur had a contemporary named Antoine Bechamp, and not a lot of people know about Bechamp. He was a little older than Pasteur, and actually Pasteur looked up to him. Bechamp was different. He was a scientist, but he was also a doctor. Pasteur was not a clinician. Bechamp had an alternative theory, almost the diametric opposite of Pasteur's, called terrain or host theory. Now, this was a health-centric theory, not a disease-centric one, and it recognized that most pathogens were actually scavengers that could only instigate what Pasteur called disease in a weakened host. For example, the flu. The flu might make a number of people symptomatic, but many people with a strong terrain, it would not result in symptoms. Bechamp viewed a dysfunction in the tissues and cells as the problem, as the origin of disease. And so this implicated something that did not need to be eliminated or suppressed, but something that needed to be restored. Now, unfortunately, in his time, he had no target. Pasteur had targets. He had these pathogens, these bacteria, these viruses as targets. Bechamp had nothing. He did not know about mitochondria back then. So he had vague recommendations, get more exercise, get more sunlight, and so on. At the end of the day, Pasteur's theory obviously won. And that leads us to today, the lifestyle disorders. Germ theory has now exploded and spilled over infectious diseases. It now pervades most of medicine, all of the non-infectious stuff too. If you look at the top 10 causes of death in New Zealand, you can actually broadly group them into four main processes. The first is diseases of the circulatory system, so heart attacks and strokes, which most heart attacks are caused by atherosclerosis, and some strokes, a number of strokes are probably related to atherosclerosis. Cancer is number two. They flip-flop. Cancer could be arguably stated as number one. Number three would be neurodegenerative disorders. 2/3 of those are due to Alzheimer's disease. And underlying the whole thing, the whole top 10, except maybe trauma and suicide, is the metabolic syndrome, defined as obesity and or being overweight centrally, with prediabetes and or type 2 diabetes and hypertension, and then lipid abnormalities. The lipid abnormalities have nothing to do with LDL. They are high triglycerides and low HDL. These are all underpinned by mitochondrial dysfunction, all of these disorders. There is absolutely extensive evidence showing that all of these disorders are underpinned by extensive mitochondrial dysfunction. A paper that came out a month ago that we authored out of this hospital goes through a number of reviews about mitochondria. If you want to look at the evidence for this, there's the URL, and do feel free to have a look. It was only published a month ago. So here's our current paradigm. This is the way we practice every day when we come to hospital. We have these lifestyle disorders which pervade our patients on the wards. You know, I think every patient this morning that I saw had the metabolic syndrome. And we see these lifestyle disorders as multiple separate disease processes. They are thought to be instigated by certain things. So, for example, the metabolic syndrome, which usually starts as a pre-diabetic sort of phenotype, is thought to be due to some kind of glucose intolerance. Atherosclerosis, we strongly attack LDL cholesterol. Cancer, we are all about genetic mutations. And of course, neurodegeneration, Alzheimer's are all about amyloid and tau being the instigators. And we think that somehow these are disease processes, and that after decades of this process, this thing just keeps going along. Eventually, the glucose intolerance results in type 2 diabetes, the LDL produces atherosclerosis, and cancer and neurodegeneration, and so on. Under this paradigm, although we sort of know better, really what we're doing every day when we come to work is we're treating health as the absence of disease. If we sort of try to get rid of the disease symptoms, then that's what we do. And so we try to suppress or eliminate these perceived pathogenic targets. Is it working? No, it's not. Because if it was working, then the lifestyle disorders would not be dominating the top 10. Here's an alternative. Here's what I think is the future. A health-centric paradigm. We have lifestyle disorders, of course, but they are actually all a single disease underpinned by mitochondria dysfunction. Which disease occurs depends on the tissue type that's most prominently damaged. You get an initial response. This is a mind flip. This is not so easy to understand. It is not a disease that we are dealing with here with these lifestyle disorders. These are responses. They are metabolic responses to the primary disease of mitochondrial dysfunction. Glucose intolerance is a response. LDL cholesterol is an intermediate. Genetic mutations are an effect or a response. And the amyloid and tau protein aggregates are an effect or response. And I'll show you why I think that. These occur over decades. If you don't fix the mitochondrial dysfunction, the responses balloon, and then you get the type 2 diabetes, atherosclerosis, cancer, neurodegeneration. This is a totally different way of looking at things, but it's crucial because it implicates that we should be treating people at the top of this little branch tree here. We should be treating the mitochondrial dysfunction. We should not be treating the sequelae of the mitochondrial dysfunction. Under this paradigm, health is defined as optimized mitochondrial function. If you have optimized mitochondrial function, the vast majority of cancers and neurodegenerative disorders and so on should not occur. So maybe we'll have more success with this kind of a paradigm working on restoring. And this appeals to me much more as a physician and as a doctor. I think physicians should be aiming to heal and restore rather than attack and suppress. This leads us to the concept of metabolic strategies. Now, metabolic strategies are greatly misunderstood things. These are methods that orchestrate a whole body metabolic state. The key word there is state, that restores mitochondrial function. That's the goal. So, ketogenic diet, for example, it's not a diet, it's a method for inducing a state. And it's not about weight loss, it's about restoring mitochondrial function. And you want to mitigate the impetus for the lifestyle disorders. Now, fasting and keto are the two main ones. They're the best, in my opinion, and the two main and most powerful ones. Fasting and keto are very similar in the state they induce. The main difference is that in fasting, the fat comes from your body fat. With the keto diet, it's fat that you eat. Otherwise, they're very similar. There are other metabolic strategies. Calorie restriction is one, specific exercise regimens. There's quite a few others, but I don't have time to talk about those today. They all induce similar mechanisms. They aim to restore mitochondrial function. Exercise has to be done in the correct manner, otherwise it can do the opposite. Fasting. Fasting is defined as a voluntary abstinence from food and drink for a specified period of time. The usual minimum is 12 hours. Starvation is involuntary. Starvation has no specified period of time. It's not starvation. There are multiple regimens. The easiest one to do is TRF, or time-restricted feeding. For example, you can have a 16-hour fasting window every day and an 8-hour eating window. You can take this up to 23-in-1. That's called OMAD, or 1 meal a day. That's a great one. So you're basically having 1 meal a day and 23 hours of fasting. There are other regimens: alternate daily fasting every second day, 5 and 2 fasting 2 days a week, and prolonged fasts, which is anything longer than 48 hours. Generally up to 3 weeks. The world record is 384 days or something like that. It's ridiculous, but no, I do not advocate that. Ketogenic diets, these are high-fat, adequate protein, low-carb diets. They limit digestible carbs to below 50 grams daily. That's a strict definition. A low-carb diet is not a keto diet necessarily. A low-carb diet limits it to 50 to 100 grams daily. Now, keto diets are not high-protein diets. They can be, but the best keto diets, in my opinion, are not high-protein diets. They're adequate protein. That's a great misconception. There are multiple regimens. Keto diets are not diets, remember. They are methods for inducing a state. So the whole argument about what's better, carnivore or vegan or vegetarian, is irrelevant with the keto diet. Now, not a lot of people understand that. I've had patients go keto carnivore, keto vegetarian. They do well, no problem. It doesn't matter. As long as you're inducing the state, the method to induce the state is there. It doesn't matter if you're carnivore or vegetarian or omnivore. You can use any ethnicity, can be adapted. Mediterranean, Thai, Indian, got people on Maori keto diets. That's a very amenable, traditional Maori diet is very amenable to keto. And you can go high fiber or no fiber. This is contentious, but you do not need fiber if you are doing this correctly. Ketones can take over a number of the functions of fiber. Metabolic strategies, again, they're an orchestrated state. It's not just a fad fasting, it's not a fad diet. They're orchestrating a state. These are powerful methods. What they do is a whole bunch of changes. I'm not going to go through the details, but they're aimed at restoring mitochondria. That's what they do. They induce an orchestra of mechanisms. They're powerful. They're not fluffy lifestyle therapies. This is the main game. There's no medication that can hope to match these if you do these correctly. So let's look at some evidence. I'm just going to briefly cover the metabolic syndrome, and I might be committing heresy here with this slide by what I'm about to say. Fasting and keto can mitigate the insulin resistance, improve the cholesterol profile, decrease hypertension, suppress inflammation, reduce the liver fat. There are dozens of interventional randomized controlled trials in people, not animals, not observational studies, that show this. They can improve and or reverse, and I do not use that word lightly, reverse the metabolic syndrome. Type 2 diabetes is in most cases reversible. If you do not believe me, please go to the paper. I've collated every single meta-analysis and study on fasting and keto in the metabolic syndrome for you to see in nice, neat tables. Okay. So again, the paper's there. It only came out a month ago, and it covers this in a more comprehensive manner than I can accommodate in this talk. I'm going to focus now on cancer and neurodegeneration. Cancer specifically with respect to the brain, because I'm a neurologist after all. Okay, cancer is characterized by a series of hallmark properties in cells followed by abnormal cell growth and spread, as we all know. And it's been over a century since we've had the somatic mutation theory, which continues to pervade our thinking. And it basically views cancer as a disease caused by nuclear mutations, as I mentioned earlier. So mutations occur, they cause cancer. Pretty simple. This is what we all learned. So the theory is that you have a quiescent cell. Mutations begin to occur in the cell nucleus. Then you get more mutations that occur in the nucleus. And after a while, some of these mutations are activated oncogenes, create a pro-cancer state, and some of these mutations are inactivated tumor suppressor genes. And the tumor suppressors normally prevent the cell from becoming cancerous. And then it's thought that you get this state of unbridled growth and proliferation. And so you can understand why we concentrate so much on the genetic properties of these cancer cells. But there are multiple inconsistencies with this theory. I only have time to go over 3 of them today. There are more in the paper, obviously. So the first inconsistency, of course, is there's rampant mitochondrial dysfunction in cancer. The somatic mutation theory just does not explain the abundant evidence that all types of human cancer express mitochondrial abnormalities. The majority of human tumors show reduced mitochondrial numbers. They exhibit disorganized cristae, often extremely so. And that's important. Cristae is where the electron transport chain is. That's where all the energy is being produced. And fusion-fission disequilibrium. Actually, mitochondria don't just sit in the cell and statically. They move around the cells, and they fuse, come together, and then they fission. They divide. And this is a normal process. There's a whole population in there. It's quite interesting. It's fascinating, actually, mitochondria dynamics, what they do. But this culminates in dysfunction, because in cancer, all these processes are messed up. So if you look at a normal mitochondria on the left, you can see it's a nice ellipsoid shape and has these beautiful, well-structured cristae. That's where the electron transport chains are. That's where almost all the energy is coming from for your cells. In glioblastoma multiforme, which is a terrible, aggressive type of brain tumor, arguably the worst you can get, here's a mitochondrion. Now, look, it's not only a slightly abnormal shape, it's got no cristae. Now, how can that mitochondrion produce energy? It seems almost impossible for it to do so, and therefore, these cancer cells, their mitochondria are very damaged. They have problems. They're in no way superior to normal cells. They're deficient. The second inconsistency, this is a very powerful one, are these Cybrid experiments. Now, the SMT cannot explain what happens when mitochondria or nuclei are transferred between normal cells and cancer cells to create what we call Cybrid cybrids, which are mitochondria from one cell, nucleus from another. Okay, now these experiments, I should say, have been done by a number of labs throughout the world, very good labs, and even some of them were trying to prove the opposite conclusion to what I'm about to show you, but they all ended up on with the same findings. Now, when you take a normal cell mitochondria, green is normal, red is not, and you combine it with a cancer cell nucleus, including all of those mutated oncogenes, according to somatic mutation theory, you should get a cancerous cell, right? The mutated cancerous nucleus with all the mutated oncogenes is there. But guess what? In almost 100% of cases, the normal state persists. Conversely, if you take a cancer cell mitochondria, cytoplasm with mitochondria, and combine that with a normal cell nucleus with its intact tumor suppressor genes, that should not become cancerous according to somatic mutation theory. But in almost all cases, the cancer state persists. This is directly opposed to the somatic mutation theory. These experiments have been around for like decades. And so it strongly implicates that the cause of cancer lies with the mitochondria, not the nucleus. Third inconsistency is this thing called the Warburg Effect, which I'm sure some people know about. The SMT does not explain the Warburg Effect. This is a near universal feature of malignant tumors. It's one of the hallmarks of cancer now, which states that even in the presence of normal oxygen levels, cancer cells ferment. Normal cells are not supposed to undergo fermentation when oxygen levels are normal. When our oxygen levels go low, then we're supposed to ferment and produce lactic acid in the process. So kind of a busy diagram, but it's not too bad. Just focus on the one on the left first. The healthy cell can use glucose or fatty acids for aerobic respiration. Healthy, normal cells are hybrid engines. They can switch to glucose or fatty acids and or the ketones which come from fatty acids, and they can do both at the same time, although that's probably not ideal. So they can use either fuel to make pyruvate, and then mitochondria uses those fuels to make a lot of ATP energy. Cancer cells are different. They upregulate their glucose intake by a factor of 10 to 100 times. It's massive. And so they become glucose-loving machines. And they really, because their mitochondria are damaged, fatty acid intake is greatly downregulated, in some cases virtually non-existent. All that glucose still makes pyruvate in the cytoplasm, but now there's so much pyruvate and these mitochondria are damaged, it becomes lactate, a lot of it. And you get a lot of ROS, or reactive oxygen species, produced in as well. So you get, that's why you get the sort of acidic tumor microenvironment, and you get all this lactate around tumors. And basically, they're just cranking out lactate. They're massively, they're massive fermentation machines, these cancer cells. And so they rely heavily on glucose. And again, SMT just doesn't really explain this very well at all. So let's look at a mitochondrial theory of cancer. You have a normal mitochondrion. Now we know that various things can cause cancer: chemical carcinogens, radiation, viruses can do it. Of course, there's a strong association with obesity and diabetes with cancer. So all of these things that can cause cancer also damage the mitochondria. The mitochondria links all these different. This is called the oncological paradox. How can so many different things cause cancer? Well, because they all damage the mitochondrion. You do this for decades, and that is what the mitochondria start to look like. The damage accumulates. They become dysfunctional. What happens then is the cell faces an energy shortage. Now, most cells will die under this condition. They just can't handle it, right? You're killing their mitochondria. But there are some cells, and this is over many cell cycles, gradually transition to the Warburg effect. They gradually upregulate their energy production in the cytoplasm, produce more lactate in the process. And what happens is they survive. Some of these cells survive, and they're the ones that are going to become the cancer cells. Now, because they have replace their primary energy source with the Warburg effect, it's okay, but they're just getting by. It's not super efficient. Their DNA repair systems need optimal mitochondrial function to work properly. So you start getting mutations still, but they're all kind of random because mutations are often occurring at random. Damage to them is occurring at random. And so it's an effect or response of the whole thing. It's not the cause of cancer, it's an effective response. So you still see it, but it's an effect. And obviously trying to treat that is not going to be successful in the vast majority of cancers, the sporadic ones, which is over 90% of cancer. And that results in your unbridled growth and proliferation. This makes a whole lot more sense to me. If this is the case, we need to restore the mitochondria. So let's look at some studies. This all started a few years ago with a unique case report. This was sort of a world first, I guess. In 2017, a young lady came and saw me with myasthenia gravis, and she had just been diagnosed with the metastatic thymoma a few weeks before. It was unresectable. She was offered a palliative chemo, but not much else could be done. So we talked about this very carefully, and she decided to go for. She wasn't keen on the chemo and perhaps having a year or so prognosis. So she pursued a combined metabolic strategy, which was these 7-day fasts every 1 or 2 months with a keto diet between the fasts for 2 years. Now it was a bumpy road, but you can see the tumor there in these 2 CT scans that is quite large. On the one on the left, that's a coronal view, and the one on the right is an axial view. And this is not a small tumor, it's the size of a small football. And there were metastases throughout the pleura in the left lung. And bumpy road, but basically at the end of 2 years, that was the tumor, 96% or so reduced in size. She did not have any chemo or radio or surgery. It was all fasting keto. And now the interpretation is not straightforward, but this is the first time a stage 4 cancer has ever had this kind of success with this kind of an approach. So it is quite encouraging, and it begs for further research, if nothing else. And she is alive and well 5 years later. I saw her just a few weeks ago. If you want to read about that, there's the report in its URL. This led us to commence a first ever case series at Waikato Hospital. Now, I decided to stick with my knitting a bit more and go for glioblastoma multiforme, that aggressive brain cancer I mentioned before. So about 2 years ago, we decided to try a similar approach in 10 patients with GBM. Now, these patients, we have a fantastic group of oncologists here who were willing to embrace this novel concept and refer some patients my way. Now, the initial patients we got were more advanced cases, and often with a recurrence, and 1 or 2 of them are actually already deemed palliative. So the toughest of the tough, advanced GBM. This is an example. This is a terrible tumor. This, you see the hyper-intense sort of white signal in the brain there. I mean, this poor guy, this was the first patient enrolled. I mean, this tumor is just terrible. It's in the middle of his brain. There's no way to resect that. I mean, that's really tough. So we recommended a combined metabolic program involving a 5 to 7 day fast every 1 to 2 months. I had very minimal involvement. This was an observational case series, basically, and the patients decided whether they would follow those recommendations, and they just did it themselves with a little bit of email help from me. There was not much I did. And basically, they successfully adhered to this program for 5 to 6 months and very mild adverse effects. So they show that it could be done even in advanced GBM cases, which is quite astounding. I was actually not sure if they could do it either. So if that was actually just recently accepted by Journal of Oncology just 2 or 3 days ago. And so I hope that comes out in 2 or 3 months. And I think it's quite an exciting thing. That's a world first. Nobody with advanced cancer of any kind of cancer has ever undergone the extensive 5 to 7 day fast that these guys did. And that has led us to our current clinical trial in GBM. So now we're conducting a clinical trial aiming for 20 patients. We already have 5 enrolled. This is newly diagnosed GBM. These are not the super advanced cases. We're getting to them. The oncologists are referring them before they start their chemo and radiation. And this is key because I think we have to combine the standard treatments with the metabolic therapies. And as an aside, I'm not against medications. We definitely need them, but we shouldn't rely on them so heavily. What I'm saying is the metabolic strategies need to become the main game to improve health. So the metabolic therapy program for these guys consists of a 5 day fast with a time-restricted keto diet. So we're combining fasting and keto. It's basically 1 or 2 meals a day, and there's a specific program outlining when you eat 1 meal, when you eat 2, and so on. And the whole thing is designed to amplify the standard treatment protocol. So chemoradiation is 6 weeks in GBM. You do a 5 day fast at the start and a 5 day fast in the middle, and the rest is that time-restricted keto diet. And then 6 months of adjuvant chemotherapy with a drug called temozolomide. You do a 5 day fast, and we time it very specifically with the 5 days of temozolomide in order to maximize the efficacy of the temozolomide and protect the normal cells. Now, there's a lot of theory there that I can't get into, but again, read the paper, the Metabolic Strategies one, and it goes into it a bit. Our objectives are to determine now feasibility and safety, of course, but also efficacy. Basically, we want to see if they live longer. That is registered here at the clinicaltrials.gov site if you want to look at the protocol. Moving away from cancer and over to neurodegeneration. Now, I see cancer and neurodegeneration as very similar disorders, okay? They're both caused by mitochondrial dysfunction. Neurodegeneration affects the neurons. It's not the neurons that get cancer in the brain. In glioblastoma multiforme, it's the glial cells. Neurons almost never get cancer, and there's a reason for that, as I hope you'll see. This is mostly Alzheimer's. Alzheimer's is associated with these protein aggregates, the amyloid plaques outside the cells, the neurofibrillary tangles inside the cell. And the problem with neurodegeneration is, at the end of the day, massive neuron loss. And for 3 decades, we've had this amyloid cascade theory and related theories like the tau theory that have seen Alzheimer's as a disease caused by these neurotoxic protein aggregates. So here's how it goes. We have a normal neuron. You get these abnormal proteins aggregating outside or inside the neuron. They are thought to be neurotoxic, so they hence interfere with neuron function. You get enough of this, and the neuron dies. That's basically what it is. That's the current model. And again, there are so many inconsistencies with this. It's somewhat remarkable, in my opinion, but I'll just mention a couple. So the first one, of course, there is rampant mitochondria dysfunction in Alzheimer's and other degenerative disorders, such as Parkinson's and Huntington's. And these precedes the plaques and tangles. Already, if something is coming before something else, then perhaps that is implicates it as the cause rather than the thing that comes later. Alzheimer's neurons show reduced mitochondria numbers, abnormal shapes, fusion-fission disequilibrium like the cancer cells, and deficient respiratory chain enzymes. It's very similar. So again, normal mitochondria with normal cristae on the left. On the right, here are some Alzheimer's cortex mitochondria. They are way too round. Their shape is messed up, and their cristae are disorganized. So again, it's very similar to Alzheimer's in that these mitochondria are structurally damaged and dysfunctional. The second inconsistency is called the Plaque deposition paradox. Now, if the amyloid cascade theory is correct and plaques cause Alzheimer's, then really you should see some kind of a correlation between the plaque load and the degree of neuron loss and the cognitive impairment, and yet the correlation is extremely tenuous. So there's a picture of some neurons with a big plaque in the middle of them there. On one hand, you have many non-demented elderly people, up sort of 40, 50% can bear very large plaque loads, and yet they have no dementia. And on the other hand, neurodegeneration can also occur in a number of patients in the absence of plaques. This is very much the opposite of what you would expect if the amyloid cascade theory were correct. So let's look at a mitochondrial theory of Alzheimer's. Again, Alzheimer's is known to be caused by a number of things, probably air pollution, probably aluminum, maybe some other heavy metals, probably, again, obesity is associated, diabetes is associated. So all these things are associated with it, and they all damage mitochondria. So it's the same thing. Damage accumulates. Mitochondria become dysfunctional after decades of abuse. The neuron faces an energy shortage. It's the same as the cancer cell, except this is a neuron. A neuron relies far more on mitochondria. A neuron is packed with mitochondria. It's extremely energy demanding. And because of that, it has limited space for glycolytic machinery. A neuron is not good at glycolysis. It's not good. So the Warburg effect relies on having the ability to go ramp up glycolysis 10 to 100 times. Neuron cannot do that. So the neuron struggles along. And then you get these proteins, these abnormal proteins building up as an effect or response of the energy crisis. Maybe they, nobody knows exactly why, of course, but maybe they are unable to be resorbed again, or maybe there's some kind of weird immune function or immune response going on. But basically, this is an effective response of the mitochondrial dysfunction, which is triggering the whole thing. And basically, because they can't go Warburg effect, neurons just die. They have no ability to go Warburg. They die. And that is neurodegeneration. So if this is true, same theory, we need to restore the mitochondria. So this led to a first-ever randomized controlled trial that we did a few years ago. This was actually in Parkinson's that we started at first. So we did a randomized trial of a keto diet in 47 patients with Parkinson's, and we compared a modified keto diet with a healthy low-fat diet. Patients on keto improved statistically, and more importantly, it was a clinically significant improvement in these non-motor symptoms, which affect the quality of life in Parkinson's more than the motor symptoms. Non-motor symptoms are things like urinary dysfunction, cognitive impairment, pain, sleep problems, that kind of thing. And interestingly, most of the improvements, the greatest ones, are seen in the most disabling symptoms. So here's a sort of a single graph of the results of the trial. The blue is the low fat and red is keto. Down is better. The low fat group improved by about 10% after 8 weeks of this, and keto improved by 40%. So it almost cut the non-motor symptoms collectively in half. That's very impressive, particularly considering levodopa has a very little effect against the non-motor symptoms that improve the most. If you want to see that one, there it is. It was published 4 years ago now. Hard to believe it was that long ago, but there's the URL. That trial led us to do another trial, a first ever in the world, again, randomized crossover trial of a keto diet, this time in Alzheimer's. So again, we're tackling these really difficult disorders, glioblastoma multiforme, Alzheimer's. You'll see the next one. We're dealing with people with Huntington's. These are the things we have to try and make better. It's not good enough to just sort of leave them in the too hard box. So this trial was published last year, much more recent, 26 patients with Alzheimer's. The randomized crossover design does not require a lot of patients. 26 patients is a very, very adequately powered statistical sample size for this kind of a design. And again, we compared a modified keto diet, this time with the participants' usual diet supplemented with healthy eating guidelines, so without sort of standard of care. And patients on keto at the end of 12 weeks, this one was, improved statistically, and again, it was clinically meaningful in daily function and quality of life. And these are the two factors of greatest importance to people with dementia. We did measure cognition too, and I'll show you the results. So blue is usual diet with recommended eating guidelines, and red is keto. and up is better in this case. So we used the ACE3 to measure cognition. You can see it declined as expected in Alzheimer's in the blue. Keto, it sort of stayed about the same, maybe improved a little bit, but it was a trend only. That was not statistically significant. There may be other reasons for that, but we couldn't get excited about it because it did not meet it. Probably the most exciting result, number one, was that in terms of function decreased on usual diet with low-fat eating recommendations, as expected, as you see the drop in the blue there. Whereas keto, it stayed about the same. That was interesting. Function seemed to sort of plateau out. And with the quality of life, this was really interesting. It sort of maybe stayed about the same with the usual diet with healthy recommended guidelines. And, you know, I think that is easy to explain. When people enter a trial and they're getting assessed every 6 weeks and they get more attention, that's sort of good for quality of life right there. So maybe that would have been part of that response. But keto improved quality of life to quite a significant extent. This is really good, not just for the patients, but for the families. Remember, Alzheimer's is often harder on the families. If you want to see that study, it was published also at this URL. Now, the success of that trial has led us to a unique case study. I wanted to do another trial in this disorder, Huntington's disease, but we had to settle for a case study because of the lockdown restrictions. So in 2021, just last year, we saw a 41-year-old man with worsening Huntington's, definite Huntington's, 47 CAG repeats. And, you know, this is a difficult, very difficult disorder characterized by motor problems, cognitive problems, and neuropsychiatric or behavioral problems. The third one is usually the worst part of it. And he was deteriorating fast over the previous 6 to 12 months. And his fiance was really worried. They were looking at care facility options. So we decided, he tried some drugs. They just didn't agree with him. He wasn't keen. So we said, okay, let's try this. So we pursued a time-restricted keto diet. So it was a 2-meal-a-day keto diet, and that's all it was. No extensive fast. His BMI was about 19. So I was really, I didn't want to have him have any more weight loss. And it was a modified keto diet, 1 year. I just thought, let's see how we go. And yeah, at the end of it, his motor symptoms improved by just over 50%, and his behavior problems, more importantly, by 50 to 100%. Now, here's the chart of the behavior problems. Remember, this is an N of 1, so don't get too excited, but it is encouraging, and it's the first case in the world of any metabolic strategy being applied to Huntington's. Apathy, down is better. So blue is at baseline, orange is at 6 months, gray is at 1 year. And you can see that apathy just slowly declined in a trend-like fashion, and it ended up being 25% of baseline. Disorientation was the next big behavior problem, and it declined by about 50%. It was eradicated by the end of the 1 year. There's no gray bar there at all. His anger and irritability were real problems for his relationships, and that they both basically declined by 50 to sort of 70%. And this, you know, basically collectively, these ones improved by 50 to 100%. Now, interestingly, anxiety there worsened at 6 months, and then it was still there a little bit at the end of 1 year. There was an unforeseen exacerbation of social factors there that I don't need to get into, but probably is explained by that. Anyways, overall great improvement, and it allowed them to get married, and now they're heading off to Europe for 6 weeks, just the two of them. This is a great difference from looking at high-level care facility options just over a year ago. That was just accepted 2 weeks ago in Frontiers in Behavioral Neuroscience, and I hope that's going to be published in the next couple months as well. So, to conclude this talk, I would submit that our modern health systems are founded on a disease-centric paradigm established 200 years ago, and we are maintaining that to this day and inappropriately applying it to non-infectious lifestyle disorders. So we currently view the metabolic syndrome, atherosclerosis, cancer, neurodegeneration as these disease processes. And so we focus on trying to eliminate these targets. We try to get rid of the amyloid plaque, we try to get rid of the mutations and so on, LDL and all the rest of it, and it's not working. A health-centric paradigm views these as a series of metabolic and hormonal responses, not diseases. We're dealing with responses in hospital most of the time. Now, there are true diseases that are non-mitochondrial based. I'm not saying everything we see is mitochondrial dysfunction, far from it. But the main ones that we're having the problems with, metabolic syndrome and so on, are. And it's all the same disease, mitochondrial dysfunction, and it's causing these sequelae, these responses. So we should be focusing on metabolic strategies to restore these people's mitochondria function. Metabolic strategies can improve or reverse the metabolic syndrome. Early results in cancer and neurodegeneration are promising, but it's really too early to draw definitive conclusions. I'm definitely not going to say that we can reverse Alzheimer's or reverse cancer. I'm not sure we can, but I think we can slow them down, and I think we really need to get more research into this area with regards to these metabolic strategies, because the early results, as I've just shown you, I hope you find them somewhat promising, if nothing else. And if this is true, look, we do have an opportunity to usher in a completely new era of health and make humanity much healthier and much better than what we currently are. Thank you very much.
Thank you, Matt.